All right, so next up here, we got the notes for section 4. We're going to be talking about traits, genes, and alleles in this section. Your objectives are to explain how there can be many versions of one gene, and then also to describe how genes influence the development of traits. Okay, so basically there's a lot of definitions in this section that we need to cover. Well, I'm going to use these terms all the time as we go on in the future, so you're going to have to know what each one of them means. So we'll pick these up and we'll work with them as we go, but we might just have to work with these more and more before you really get them drilled into your head. So your first definition is a gene, and a gene is a piece of DNA that stores instructions to make a certain protein, and that protein is whatever it is that's going to lead to that certain trait. So you got DNA that makes you you, and that's going to be a gene. Right? Alleles are any different forms of a gene. Right? So although we could just be talking about flower color in that example of that experiment of Mendel's, and you have purple and white, right? There are different forms of a gene, and those are the different traits. So when I'm talking alleles, I'm not talking about a different gene, but I am talking about a different form of that gene. So I will be talking about flower color, but it's a different color, right? Or in an example that's more relevant to you, right? You have a certain eye color that's different from somebody else's, right? There's blue, green, brown, hazel, all sorts of different colors of eyes, and those are going to be different alleles for that gene. So different forms of that gene that code for different traits, but they're really similar traits, just different variations of that same thing. Okay, the next term is homozygous. That would be any organisms with two of the same allele. So those would be the parents from Mendel's experiment, right? One parent always was purple, so they only had that purple allele. Whereas the other parent was always white, so they must have had two of the white alleles, no purple alleles. Heterozygous is an organism with two different alleles. And that's what we think is going on in that second generation of Mendel's experiment, right? Any flower with a purple and a white allele is just going to be purple because the purple happens to be dominant to the white. It can mask the white allele. But that organism with two different alleles had the ability to pass on the purple or the white, so white could show up again in that next generation. So homozygous, you have two of the same alleles. Heterozygous, you have two different alleles. Only one color is going to show up when you're talking about flower colors. You're only really going to have one version of that trait, but you might have two different alleles for two different things. Okay, the genome is all of an organism's genetic material combined, so all of your DNA combined is your genome. Genotype is a specified combination of alleles in an organism, whether it be homozygous or heterozygous, right? Knowing exactly what alleles you have is going to be kind of important. So different genotypes, again, you can be homozygous, have two of the same allele for two of those dominant alleles, as we'll see later, or you could be heterozygous, have two different alleles. Makes a pretty big difference. Phenotype is the trait shown by an organism. So although you might have a purple and a white allele, if only purple shows up, that's the phenotype. What do you see? So the genotype is not actually something that we can see because we can't tell the difference between the purple flower in the parental generation here and the purple flower and the F1 generation, they look the same. They're both purple. But one is homozygous, the other is heterozygous. That's the genotype. The phenotype is just the color that you see. It's purple or it's white. Okay, and then that brings us into dominant and recessive. If you are heterozygous, right, if you have two different alleles, the allele that's actually expressed is your dominant trait. Right, so we're going to use capital letters for those, as you've already seen with my example above. That capital P is for purple. Lowercase p is actually the recessive. When I had those two things together in that F1 generation, I still had a purple flower. I still had that purple phenotype, but it was that heterozygous genotype. And purple showed up because it was dominant. The only way that a flower could be white is if you had two recessive alleles. So in that third generation, 1 in every 4 flowers was white, and the only way to be white is to have two lowercase recessive alleles. Right? We're going to use lowercase because it's convenient, but when you have two recessive alleles, that's the only way that you can have that recessive trait. Because again, if you're heterozygous, you have one big and one little, that's going to lead to the dominant trait that comes through. So the only way to be white in our example was to have two recessive, two lowercase p's. Okay, so again, consider the following, the flower example from before. Remember the purple allele was dominant, so that capital P, white was recessive, lowercase p. So again, in that first generation, the parents were either purple or they were white, and they were purebred. So we knew that they were going to be homozygous. One group of flowers was always purple, the other was always white. So you have those two purebred lines that were being mixed. That next generation, because the purple flowers only had that dominant allele to give and the white flowers only had the recessive allele to give, that second generation were all heterozygous. And because they were heterozygous and purple's dominant, purple's the trait that showed up. But once I cross those flowers, or let them self-pollinate, which is another way of just letting them cross, then that white trait can show up again. Because both parents, in this case, have a recessive allele to give. If they both did give that recessive allele, you could have that white trait showing up again. In that third generation, though, we would have had all different possibilities, right? Some of the new offspring would have been homozygous dominant. Some of them would have been heterozygous, and some of them would have been homozygous recessive. So I would have had a mixture of different things. Obviously, just from what we can see here alone, there'd be more purple than white. And it turns out there were actually 3 purples for every white flower, which made a big difference when we're talking about meiosis and how that works, and Punnett squares here in the future. So again, this is just the same thing written out. In that P generation, that parental generation, the purebred purple flower was homozygous dominant. That purebred white flower was homozygous recessive. And the F1 generation, all of the flowers were heterozygous. And because they're heterozygous and have that dominant allele, they're all going to be purple. And then in that F2 generation, some of the flowers were purple, some of them were white. That white showed up again. The purple flowers could have been homozygous dominant, or they could have been heterozygous. We're not really sure. You can't tell. All you know is that they're purple, so you know their phenotype, but you don't necessarily know their genotype. It could have two dominant alleles. It might have a dominant and a recessive. But all of the white flowers for sure had to have been homozygous recessive. So you can see white is the phenotype, but you also know the genotype. It would have had to have two recessive alleles. All right, so hopefully that covers it.